Today we're diving into the fascinating world of controlled drug release from porous microcapsules and microgels. This is a critical technology in modern pharmaceutical science that allows us to deliver drugs at precisely controlled rates over extended periods. First, let's understand what we're dealing with. Microcapsules and microgels are tiny spherical structures, typically ranging from 1 to 1,000 micrometers in diameter. That's roughly the size of a red blood cell, up to the width of a human hair. Microcapsules have a core shell structure, with the drug stored in a central reservoir surrounded by a polymer membrane. Microgels, on the other hand, are three-dimensional networks where drug molecules are dispersed throughout a cross-linked polymer matrix. You might ask, why do we need controlled release in the first place? Great question. Traditional drug delivery has major limitations. When you take a traditional pill, the drug is absorbed quickly, creating a spike in blood concentration. This spike might exceed the toxic level, then rapidly decline below the therapeutic window. This means periods where the drug is either too concentrated or ineffective. This graph shows the problem. The red line is the toxic level, and the green line marks the minimum effective concentration. With traditional pills, you get dangerous peaks and ineffective troughs. Controlled release systems maintain drug concentration within the therapeutic window for extended periods. This improves efficacy, reduces side effects, and increases patient compliance because you don't need to take pills multiple times per day. Now let's explore the three fundamental mechanisms that govern drug release from these systems: diffusion, degradation, and swelling. Each mechanism follows distinct kinetics and can be engineered to achieve specific release profiles. Diffusion is the movement of drug molecules from regions of high concentration to low concentration. This is governed by Fick's laws of diffusion, which are fundamental to understanding release kinetics. Fick's first law states that the flux J, that's the amount of drug crossing a unit area per unit time, equals negative d times the concentration gradient d, c over dx. D is the diffusion coefficient, measured in square centimeters per second. The diffusion coefficient depends on several factors: the size of the drug molecule, the porosity of the polymer matrix, and the tortuosity of the diffusion pathways. Smaller molecules diffuse faster, and more porous materials allow quicker release. Here you can see drug molecules in red, diffusing from left to right, from high concentration to low concentration. The green arrows show the net direction of molecular movement driven by the concentration gradient. Let's do a quick back-of-envelope calculation to see how long it takes for a drug to diffuse through a polymer membrane. This will give us intuition about timescales. The characteristic diffusion time T is approximately L squared divided by D, where L is the diffusion distance. This comes from dimensional analysis of the diffusion equation. Let's say we have a membrane that's 100 micrometers thick, which is 0.01 centimeters. And let's assume a typical diffusion coefficient of 10 to the negative 7 square centimeters per second for a small drug molecule and a hydrogel. Plugging in the numbers, 0.01 squared is 10 to the negative 4. Divided by 10 to the negative 7 gives us 10 to the third seconds, which is about 17 minutes. So the drug takes roughly 17 minutes to diffuse through this membrane. Notice that time scales with the square of the thickness. If you double the membrane thickness, the release time quadruples. This is why membrane design is so critical for controlling release rates. For diffusion-controlled release from a sphere or slab, the cumulative amount of drug released often follows the square root of time law, especially in the early stages. The mass released at time t, denoted m sub t, is proportional to the square root of t. The proportionality constant k depends on the diffusion coefficient, drug solubility, and geometry of the system. In this plot, the cumulative drug release follows a square root relationship with time. You can see the curve has that characteristic shape, rapid initial release that gradually slows down as the concentration gradient decreases. Now let's discuss degradation-controlled release. In this mechanism, the polymer matrix or capsule shell degrades over time, exposing drug molecules and allowing them to escape. This is particularly useful for sustained release over days to months. Common biodegradable polymers include polylactic coglycolic acid, or PLGA, polycaprolactone, and polylactic acid. These polymers hydrolyze in physiological conditions, breaking down into biocompatible products that the body can eliminate. Here's a visual representation. On day 0, the microgel is intact with drug molecules trapped inside, shown in yellow. By day 7, the polymer has partially degraded, creating pathways for drug to escape, shown in red, leaving the matrix. Polymer degradation can follow different kinetic models. First-order kinetics means the degradation rate is proportional to the amount of polymer remaining. Zero-order means degradation proceeds at a constant rate regardless of polymer mass. For first-order degradation, dm over dt equals negative k sub d times m, where m is the polymer mass and k sub d is the degradation rate constant. This gives exponential decay. Integrating this gives m of t equals m0 times e to the negative k dt. The polymer mass decreases exponentially with time. Let's calculate how long it takes for a PLGA microcapsule to degrade to half its original mass. This is important for designing release systems with specific durations. The half-life is natural log of 2, divided by the degradation rate constant Kd. For PLGA with a 50-50 ratio of lactic to glycolic acid, Kd is typically around 0.01 per day at body temperature. So T half equals 0.693 divided by 0.01, which gives us about 69 days. This means the PLGA microcapsule loses half its mass in about 2 months, allowing sustained drug release over this period. By changing the ratio of lactic to glycolic acid in PLGA, you can tune the degradation rate. More glycolic acid makes it degrade faster because glycolic acid is more hydrophilic and water penetrates more easily. The third major mechanism is swelling-controlled release. This is particularly important for hydrogels, which are cross-linked polymer networks that can absorb large amounts of water and swell significantly. Some hydrogels can absorb water and swell to 10 to 1,000 times their dry weight. This dramatic volume change opens up the polymer network and allows drug molecules to diffuse out much more easily. Here you see a microgel particle swelling. In the dry state, it's small and compact with radius 100 micrometers. Upon water absorption, it swells to 250 micrometers radius. The volume increases by a factor of 15. The rate and extent of swelling depend on the cross-link density of the polymer network. More cross-links restrict swelling. Also, pH-responsive polymers can swell dramatically at specific pH values, making them useful for intestinal drug delivery. The swelling ratio Q is defined as V swollen divided by V dry. This tells us how many times the volume increases. For our example, Q equals 15.6. As the hydrogel swells, the distance between polymer chains increases. This mesh size determines how easily drug molecules can move through the network. The mesh size xi is approximately phi to the negative 1/3 power times L, where phi is the polymer volume fraction and L is the bond length between crosslinks. As the gel swells, phi decreases and mesh size increases. Here's the beautiful part. As the gel swells, the mesh size increases, which increases the diffusion coefficient, which causes faster drug release. This creates a self-accelerating release profile that's different from simple diffusion. In real pharmaceutical systems, multiple mechanisms often operate simultaneously. Understanding how they interact is crucial for designing optimal release profiles. Consider a PLGA microsphere. Initially, drug release is controlled by diffusion through the polymer matrix. As the polymer degrades over weeks, porosity increases, accelerating diffusion. Finally, the matrix erodes completely, releasing any remaining drug in a burst. This triphasic release profile shows all 3 phases: the initial slow release from diffusion, the accelerating middle phase as degradation increases porosity, and the final burst as the matrix erodes completely. To quantitatively describe release kinetics, pharmaceutical scientists use semi-empirical models. The most widely used is the Pepys power law equation. Mt over m infinity equals k times t to the n. Here, mt is the amount released at time t, m infinity is the total amount released at infinite time, k is a constant, and n is the release exponent that tells us the mechanism. The value of n is diagnostic. For spherical particles, n equals 0.43 indicates Fickian diffusion. If n equals 0.85, it's case 2 transport, meaning swelling-controlled release. Values between indicate anomalous transport, where multiple mechanisms operate. For n equals 0.43, we have Fickian diffusion. The release rate decreases with time as the concentration gradient diminishes. For n equals 0.85, swelling controls release. The polymer network relaxation is the rate-limiting step. When n falls between these values, both diffusion and swelling contribute to the release mechanism. This is called anomalous transport. Let's examine a real-world application, designing a glucose-responsive insulin delivery system using pH-sensitive microgels. This is cutting-edge research for treating diabetes. The idea is elegant. When blood glucose levels rise, local pH changes slightly. This triggers pH-sensitive microgels to swell, increasing mesh size and releasing insulin. As glucose normalizes, the gel contracts, slowing insulin release. It's a self-regulating system. Here's the glucose-responsive system. At low glucose, the gel is contracted. When glucose rises and pH drops to 7.2, the gel swells and releases insulin shown by the green arrows. It's a beautiful example of smart drug delivery. Let's do a practical calculation. How much insulin must be loaded into microgels to maintain therapeutic levels for 24 hours? A typical type 1 diabetic requires about 40 units of insulin per day. 1 unit of insulin is 35 micrograms, so we need 1,400 micrograms per day. That's 40 times 35, which equals 1,400 micrograms of insulin per day. Let's say each microgel can hold 0.1 micrograms of insulin. How many microgels do we need? 1,400 divided by 0.1 equals 14,000 microgels. That sounds like a lot, but remember these are tiny structures, only tens of micrometers in diameter. Let's calculate the total volume. If each microgel has a diameter of 50 micrometers, that's a radius of 25 micrometers, or 0.0025 centimeters. The volume of one sphere is 4/3 pi r cubed. That's 4/3 pi times 0.0025 cubed, which equals 6.5 times 10 to the negative 8 cubic centimeters per microgel. Total volume is 14,000 times 6.5 times 10 to the negative 8, which equals about 9.1 times 10 to the negative 4 cubic centimeters. That's less than 1 microliter. Incredibly small. This minuscule volume means the entire daily insulin dose could be delivered in a single subcutaneous injection of less than 1 microliter. This demonstrates the power of microencapsulation technology. Now let's discuss how pharmaceutical engineers design specific release profiles. There are several key parameters you can tune to achieve desired kinetics. Higher molecular weight polymers degrade more slowly, extending release duration. For PLGA, molecular weights range from 10,000 to 200,000 Daltons, giving degradation times from weeks to over a year. Higher cross-link density restricts swelling and slows diffusion. By controlling the ratio of cross-linking agent to polymer, you can tune the mesh size and release rate over a wide range. The amount of drug loaded and how it's distributed affect release. Surface-loaded drugs show burst release, while uniformly distributed drugs give more sustained release following Fickian kinetics. Remember our diffusion timescales with length squared. Smaller particles release faster. Also, geometry matters. Cylinders, disks, and spheres have different surface area to volume ratios affecting release kinetics. Here you see how geometry affects release. Small spheres release quickly because of short diffusion distances. Large spheres are slower. Disk shapes have high surface area to volume ratios, also promoting faster release from the large flat surfaces. Here's an important question. If constant, zero-order release is ideal for maintaining steady drug levels, why don't we see it more often in practice? Good question. True zero-order kinetics requires very specific conditions that are hard to achieve. You need constant surface area, which means degradation must exactly compensate for size reduction. Or you need a rate-limiting membrane with perfect properties. In practice, most controlled release systems show approximately zero-order kinetics over a limited time window, perhaps between 20 and 60% drug release. Outside this range, you see deviations due to changing geometry and depletion effects. Reservoir systems with rate-controlling membranes come closest to true zero-order release. As long as the reservoir maintains saturation conditions and the membrane doesn't change, you get constant flux described by Fick's first law. Let's briefly explore some advanced topics in controlled release. Modern research focuses on stimuli-responsive or smart drug delivery systems that respond to biological signals. Poly-N-isopropyl acrylamide, or PNIPAM, undergoes a sharp phase transition at 32 degrees Celsius. Below this temperature, it's hydrophilic and swollen. Above it becomes hydrophobic and collapses. This can trigger drug release. Some systems use peptide cross-links that are cleaved by disease-specific enzymes. For example, matrix metalloproteinases are overexpressed in tumors. Microgels with MMP cleavable cross-links selectively release drugs at tumor sites. Incorporating magnetic nanoparticles allows external control. Applying an alternating magnetic field heats the nanoparticles, raising local temperature and triggering release from temperature-sensitive polymers. This enables on-demand, spatially controlled delivery. Controlled release from microcapsules and microgels isn't just academic research. There are numerous FDA-approved products on the market using these technologies. Lupron Depot uses PLGA microspheres to deliver leuprolide acetate over 1, 3, or 4 months. This treats advanced prostate cancer with just 4 injections per year instead of daily pills. Patient compliance improved dramatically. Risperdal Consta delivers risperidone from PLGA microspheres over 2 weeks. This long-acting injectable helps schizophrenia patients who struggle with daily medication regimens. Ozurdex is a biodegradable implant that releases dexamethasone into the eye over several months to treat macular edema. The PLGA rod slowly degrades, providing sustained anti-inflammatory effects without repeated injections. Let's summarize the key principles we've covered about controlled drug release from porous microcapsules and microgels. The 3 fundamental mechanisms are diffusion governed by fixed laws, polymer degradation following first-order or zero-order kinetics, and swelling that changes mesh size and diffusion coefficients. For diffusion-controlled systems, remember that time scales with the square of diffusion distance. This makes particle size a powerful design parameter. The choice of polymer is critical. Polymer molecular weight, composition, and cross-link density determine degradation rates and swelling responses that directly control release kinetics. Semi-empirical models like the Peppa's power law equation allow us to characterize release mechanisms and predict behavior. The release exponent n is diagnostic of the controlling mechanism. In practice, multiple mechanisms operate together, creating complex release profiles. Understanding these interactions is essential for rational design of drug delivery systems. Advanced stimuli-responsive systems represent the future of drug delivery. These smart materials respond to temperature, pH, enzymes, or external fields to provide on-demand, site-specific drug release. Looking forward, controlled release technology is moving toward personalized medicine and highly targeted delivery. Imagine systems that release drugs only at disease sites, respond to individual patient metabolism, and adjust dosing in real time based on biosensor feedback. Thank you for exploring the fascinating world of controlled drug release from microcapsules and microgels. This technology is revolutionizing medicine, improving patient outcomes, and enabling new therapeutic strategies that were impossible just decades ago.